Welcome to Bio 211D Plant Physiology, Unit 4, Session 3, Oxidative Phosphorylation and Photophosphorylation. By the close of this session, you should be able to define photorespiration, explain conditions which favor photorespiration, explain oxidative phosphorylation, describe the processes in photophosphorylation, compare oxidative and photophosphorylation, explain the benefits of photophosphorylation and describe mechanisms used by plants to control photophosphorylation. Before we look at the details of photophosphorylation, let us first understand what photorespiration is. From the name, we can deduce that it is respiration that takes place or happens in light. This is not far from the truth since it occurs during photosynthesis. Photorespiration is also known as the oxidative photosynthetic carbon cycle or C2 photosynthesis. It is a process in plant metabolism which tries to reduce the consequences of a wasteful oxygenation reaction by the enzyme Rubisco. The desired reaction is the addition of carbon dioxide to ribulose biphosphate. This is also referred to as carboxylation of ribulose biphosphate, which is a key step in the Calvin-Benson cycle you have already seen in photosynthesis. However, about 25% of reactions by Rubisco instead add oxygen to ribulose biphosphate. This is what is referred to as oxygenation. Oxygenation of ribulose biphosphate results in the production of a metabolite that cannot be used within the Calvin-Benson cycle. Thus, the oxygen of ribulose biphosphate reduces efficiency of photosynthesis. That is, the output or production of glucose is reduced. This reduction in glucose production can be as high as 25% in C3 plants such as rice or dicot. Photorespiration involves a complex network of enzyme reactions that exchange metabolites between chloroplast, leaf, peroxisomes, and mitochondria. However, we shall not go into the details of this. These details shall be discussed at a higher level. The oxygenation reaction of Rubisco is a wasteful process because 3-phosphoglycerate is created at a reduced rate and at higher metabolic cost as well as compared with the ribulose biphosphate carboxylate activity. While photorespiratory carbon cycling results in the formation of G3P eventually, there is still a net loss of carbon. This loss is about 25% of carbon fixed by photosynthesis, which is re-released as carbon dioxide and nitrogen as ammonia. The ammonia produced is toxic to the plant. Therefore, the ammonia must be detoxified at a huge cost to the cell. Photorespiration also results in the loss of 1 ATP and 1 NADP each. The entire process of photorespiration refers only to the metabolic network which acts to reduce the product of the oxygenation reaction, that is phosphoglycolate. Now let us briefly see how the process of photorespiration happens. Addition of molecular oxygen to ribulose 1,5-biphosphate produces 3-phosphoglycerate PGA and 2-phosphoglycolate 2PG or PG as shown in the equation below. This is referred to as the oxidative photosynthetic carbon cycle reaction catalyzed by ribulose biphosphate oxygenase activity. So ribulose biphosphate plus oxygen given as phosphoglycolate plus 3-phosphoglycerate plus 2 molecules of hydrogen. So remember that PGA is a normal product of carboxylation which enters the Calvin cycle as we have seen in the dark phase of photosynthesis. The phosphoglycolate produced by this reaction or process, however, inhibits some enzymes involved in photosynthetic carbon fixation. That is how PG is described as an inhibitor of photosynthesis. PG is relatively difficult to recycle. However, in higher plants, it goes through a series of reactions in the peroxisome, mitochondria, and then again in the peroxisome where it is converted into glycerate finally. The glycerate produced then re-enters the chloroplast and by the same transporter that exports glycolate. 1 molecule of ATP is used for the conversion of glycerate to 3-phosphoglycerate. That is, a molecule of ATP is used to phosphorylate glycerate and the phosphorylation produces 3-PGA. This takes place within the chloroplast, and the EGA is then free to re-enter the carbon cycle. Photorespiration may also lead to the production of hydrogen peroxide in the peroxisome. Hydrogen peroxide is a dangerously strong oxidant which must be immediately broken into water and oxygen by an enzyme called catalase. Photorespiration rates are increased by lowered carbon dioxide or increased oxygen. High temperatures. So remember that carbon dioxide is a substrate for photosynthesis. Thus, its availability will influence photorespiration since carbon dioxide and molecular oxygen compete for repeat score. This simply implies that the factors which can influence substrate availability, thus carbon dioxide and oxygen, include the atmospheric abundance of the two gases, the supply of the gases to the site of fixation, that is, whether the stomata are opened or closed, and the length of the liquid phase of the gases. So in other words, the extent to which these gases have to diffuse through water in order to reach their reaction site can also affect the availability to compete for Rubisco. So for instance, as we shall see in Unit 6, when the stomata are closed to prevent water loss during drought condition, it limits the carbon dioxide supply. At the same time, oxygen production within the leaf will continue. What do you think will happen at this point? Oxygen concentration in the leaf will be higher than that of the carbon dioxide. So meaning the oxygen molecules will react with Rubisco, leading to an increase in photorespiration. So with increase in carbon dioxide concentration, however, the reverse will be true. Carbon dioxide concentrations have been increasing over the years. Therefore, it is believed that this may reduce photorespiration in vascular plants. So when temperatures increase beyond certain levels, Rubisco is less able to discriminate between carbon dioxide and oxygen. This is because an intermediate metabolite called NADO is less stable. High temperatures or abnormally high temperatures also reduce the solubility of carbon dioxide, much more over that of molecular oxygen. This then leads to reduction of carbon dioxide concentration compared to that of oxygen in the chloroplast. Consequently, photorespiration increases. Photorespiration may serve a good reason after all. Photorespiration may serve as protection against excessive sunlight and may be necessary for assimilation of nitrate from soil, among others. Even though photosynthesis may be reduced by as much as 25%, photorespiration may be used as a mechanism to dissipate excess energy at high irradiance level. Plant mechanisms usually operate to optimize the rate of photosynthesis. Therefore, along with other non-photosynthetic thermal dissipation methods, plants can use photorespiration as a method of photoprotection. In vascular plants, reducing photorespiration may not result in increased growth rates. It is rather thought that photorespiration may be necessary for the assimilation of nitrate from soil. Thus, a reduction in photorespiration by genetic engineering or increasing atmospheric carbon dioxide due to fossil fuel burning may not benefit plants. Photorespiration increases availability of NADH, which is required for the conversion of nitrate to nitrite. It has also been proved that certain nitrite transporters also transport bicarbonate, and elevated carbon dioxide has been shown to suppress nitrite transport into chloroplast. That is why a reduction in photorespiration may not benefit plants. The photorespiration pathway influences a wide range of processes from bioenergetics, photosystem to function, and carbon metabolism to nitrogen assimilation and respiration. You see how important some processes that seem not beneficial are important in vascular plants. The photorespiratory pathway is a major source of hydrogen peroxide in photosynthetic cells. Though hydrogen peroxide production and pyridine nucleotide interactions, photorespiration makes a major contribution to cellular redox homeostasis. In so doing, it influences multiple signaling pathways, particularly those that regulate plant hormonal responses, controlling growth, environmental and defense responses, and programmed cell depth. Photorespiration may also serve as a safety valve, preventing the excess of reductive potential coming from an over-reduced NADPH pool from reacting with oxygen and producing free radicals. Free radicals are reactive oxygen species that can damage the metabolic functions of the cell by subsequent oxidation of membrane lipids, proteins, or nucleotides. Some plant species, as well as some algae, have mechanisms for reducing the uptake of molecular oxygen by Robisco. These mechanisms are referred to as carbon concentrating mechanisms. They are used to increase the concentration of internal carbon dioxide so that Robisco is less likely to produce glycolates through reaction with oxygen. That is, the oxygenation reaction of Robisco is reduced when the carbon dioxide concentration is highly internally. These mechanisms include biochemical carbon concentrating mechanisms such as the C4 and crassulation acid metabolism pathways (CM) and biophysical carbon concentrating mechanisms. Biochemical carbon concentrating mechanisms. These CCMs concentrate carbon dioxide in one temporal or spatial region through metabolite exchange. As stated earlier, these mechanisms are the C4 and the crassulation acid metabolism CAM photosynthetic pathways. Both use an enzyme called phosphoenol pyruvate carboxylate to add carbon dioxide to a 3-carbon sugar to produce a 4-carbon compound. Pep-C is faster than Robisco and is more selective for carbon dioxide. As stated earlier, millet, sorghum, maize, and sugarcane plants use the C4 pathway, thus minimizing photorespiration. What C4 plants do is they capture carbon dioxide in their mesophyll cells using the enzyme called phosphoenol pyruvate carboxylase. This enzyme catalyzes the combination of carbon dioxide with a compound called phosphoenol pyruvate (PEP). The resultant compound formed is called oxaloacetate. This oxaloacetate is then converted to malate. The malate formed is transported into the bundle sheath cells, which is the site of carbon dioxide fixation by Robisco. So the concentration of oxygen is low in the bundle sheath cells, hence photorespiration is avoided. So in the bundle sheath cells, carbon dioxide is removed from the malate and then combined with ribulose biphosphate by Robisco in the usual way, and the carving cycle proceeds as normal. So it has been estimated that the concentration of carbon dioxide in the bundle sheet ranges between 10 to 20-fold higher than the concentration in the mesophyll cells. So the ability to avoid photorespiration makes C4 plants hardier than other plants in dry and hot environments, where stomata of leaves are often closed and internal carbon dioxide levels are low. Under these conditions, photorespiration occurs in C4 plants, but at a much reduced level compared with C3 plants such as rice and pepper, just to mention a few in the same conditions. You should now understand why millet, sorghum, and maize are able to give high yields under the dry, hot savanna zones of Ghana, such as northern Ghana. You have seen cactus plants or plants growing in desert or desert-like conditions, such as by the seashore or dry savanna. If not referred to the model on morphology and anatomy of vascular plants, these plants are referred to as crassulation acid metabolism plants, that's CAM plants. Such plants as cactus and other succulent plants also use the enzyme PEP carboxylase to capture carbon dioxide. However, they only capture carbon dioxide at night. The CAM pathway allows plants to conduct most of their gas exchange in the cooler nighttime air, storing carbon dioxide in a 4-carbon sugar, which can be released to the photosynthesizing cells during the day. This allows CAM plants to reduce transpiration and consequently water loss by closing their stomata during the day. This also enables them to survive the harsh weather conditions in which they live. CAM plants usually display other water-saving characteristics such as thick cuticles stomata with small apertures as discussed in Adaptations to Water Loss in Unit 6. This type of carbon concentrating mechanism relies on a contained compartment within the cell into which carbon dioxide is transported. In this compartment, also, Ribisco is highly abundant and readily available to make use of the carbon dioxide. In many species, biophysical CCMs are only induced under low carbon dioxide concentrations. Oxidative phosphorylation. We can now look at oxidative and phosphorylation, photophosphorylation. But first, oxidative phosphorylation. We have already discussed the Krebs cycle and the electron transport chain in Unit 2. All the processes in aerobic respiration that involve phosphorylation to produce ATP are referred to as oxidative phosphorylation since oxygen is required in aerobic respiration. So oxidative phosphorylation occurs in the mitochondria and molecular oxygen reduced to water using electrons donated by nicotinamide adenine dinucleotide, reduced nicotinamide adenine dinucleotide, or FADH2. Albert Leninger discovered that mitochondria are the sites for oxidative phosphorylation in 1948. Remember the structure and function of mitochondrial cells. So in the Krebs cycle, it produces reduced NAD, that is nicotinamide adenine dinucleotide, and reduced FAD. The NADH and FADH2 produced by the Krebs cycle are reoxidized, and the energy released is used to produce ATP. This process is referred to as oxidative phosphorylation since they donate the hydrogen ions. That is, oxidative phosphorylation is ATP synthesis linked to the oxidation of NADH and FADH2. This is done by electron transport through the respiratory chain. How does it occur? Here, unlike substrate level phosphorylation, it does not involve phosphorylated chemical intermediates. It involves the chemiosmotic hypothesis proposed by Peter Mitchell in 1961. In this mechanism, energy released by electron transport is used to pump hydrogen ions out of the mitochondria, thus creating an electrochemical proton gradient. The protons flow back into the mitochondrion through an ATP synthase located in the inner mitochondrial membrane, and this drives ATP synthesis. As studied earlier in Unit 2, 3 ATP molecules are synthesized per each NADH oxidized, while 2 ATP molecules are synthesized per each FADH2 molecule oxidized. Photophosphorylation. As you have seen in photosynthesis, ATP is also produced at some stages in the light phase. Therefore, photosynthetic phosphorylation is also called photophosphorylation. It is the process where a phosphate group is transferred to ADP in order to synthesize the energy-rich ATP molecule with the aid of sunlight or solar radiation as external energy source. According to the chemiosmotic hypothesis mechanism proposed by Peter Mitchell, the ATP is synthesized on ATPase complexes located on the non-appressed portions of thylakoid membranes, particularly toward margins. This is made up of non-cyclic and cyclic as shown in the diagram. Just as for oxidative phosphorylation, hydrogen protons accumulate in the thylakoid space due to the splitting of water and transport between PQH2 to cytochrome F during the photosynthetic electron transport. The increase in the concentration of hydrogen protons in the thylakoid space results in increase in the proton gradient. The downflow of protons from high to low concentration along hydrogen concentration gradient through the ATPase complex provides the energy that drives an ATPase synthase enzyme to synthesize ATP from ADP plus E. The electrons lost by the reaction center P68 of photosystem 2 are taken up by the reaction center P700 of photosystem 1 and do not get back to photosystem 680. Hence, it is unidirectional, or the electron flow is in one direction only. That is why it is called non-cyclic phosphorylation. The electrons pass through the primary acceptor, plastoquinone PQ, cytochrome complex, plastoquinone PC, and finally to P700. The electrons released by P700 taken up by primary acceptor are ultimately passed to NADP. The electrons combine with protons or hydrogen ions and reduce NADP to NADPH2. Note the difference here to that for oxidative phosphorylation. The hydrogen ions are made available by splitting up of water. Thus, non-cyclic photophosphorylation needs a constant supply of water molecules to constantly provide protons and evolve molecular oxygen. The net result of non-cyclic phosphorylation is the formation of oxygen, NADPH, and ATP molecules. Oxygen is produced as a waste product of photosynthesis so that humans and other animals can have oxygen for cellular respiration. This is one of the reasons for the saying that if the last tree dies, the last man dies. So this is one of the diagrams that we've already discussed. You have light going through photosystem 2, plastoquinone, the cytochrome, then we have it moves to the photosystem 1, then the processes goes on until everything is reached. Cyclic photophosphorylation. In cyclic photophosphorylation, the electrons released by P700 of photosystem 1 in the presence of light are taken up by the primary acceptor and are then passed on to ferrodoxin, plastoquinone, cytochrome complex, plastoquinone, and finally back to the P700. Note that here electrons return or flow back to the same molecule they originated from after cyclic movement. Cyclic photophosphorylation also results in the formation of ATP molecules, just as in non-cyclic photophosphorylation. So the diagram is as discussed. You have chlorophyll, you have light, excited stage, the pathways of electrons through the various photosystems. So the cyclic photophosphorylation, also the electrons move downhill in the electron transport chain. Hence, they also lose potential energy, and the energy released is used to form ATP molecules in the same way as in mitochondria during respiration. Here, electrons from photosystem 1 are not passed to NADP from the electron acceptor. Rather, the electrons are transferred back to P700. This downhill movement of electrons from an electron acceptor to P700 results in the formation of ATP, and this is referred to as a cyclic photophosphorylation. Please note that oxygen and NADPH2 are not formed during cycle photophosphorylation. We can therefore state that in non-cyclic photophosphorylation, electrons do return to the same molecule. Hence, in the flow is unidirectional. The first electron donor is water. It involves both photosystem 1 and 2. The final electron acceptor is NADP, and the net products are ATP, NADPH, and oxygen. For cyclic photophosphorylation, however, electrons are as follows. So here the diagram, that is how it goes through. So oxidative phosphorylation is spontaneous and occurs in the mitochondria, and molecular oxygen reduced to water using electrons donated by NADH and FADH2. Photophosphorylation just the reverse, where water oxidized to oxygen with electrons accepted by NADP plus, not spontaneous. Energy must be supplied by light, and it occurs in the chloroplast. However, both processes are highly efficient and similar in mechanism. Both phosphorylation and photophosphorylation have these same characteristics, as both involve flow of electrons through membrane-bound carriers. Downhill electron flow is used to transport protons through a proton-impermeable membrane against a concentration gradient. So energy is conserved not in chemical bonds, but in an electrochemical potential. Protons are then allowed to flow down their concentration gradient through specific channels, and this flow releases energy which is used to synthesize ATP. So in review, photorespiration was defined and the conditions that favor the process were explained. We also talked about oxidative phosphorylation and then described the processes in photophosphorylation. So the differences and or similarities between oxidative phosphorylation and photophosphorylation were considered. The importance of photophosphorylation to plants and the mechanisms used by plants to control photophosphorylation.